Welcome back. You guys know the drill right by now, so let's get into it. This is the third week of distance learning. So the focus for this week, since we went over the whole process last week of transcription and translation, with a bigger focus on transcription, I want to spend a good chunk, and by good chunk, I mean all of this week, just practicing translation, because this is the part where I see a lot of people get confused. This is the part where I see a lot of people um, make easy mistakes that are understandable. So I really want to take some time to practice this, and then depending on how this goes, we may spend some more time on it next week, we may not. I'm going to try and limit it to this week so we don't spend too much time on it. But anyway, let's get into it. So, quick reminder of things we've talked about so far because they'll be important as we go forward. Translation is the process of turning the mRNA that we formed in transcription into a usable protein. So if you remember from last week, I gave you a DNA sequence, and you would turn the DNA sequence into an mRNA sequence. It's the same thing as replication. You just flip the letters, with the major difference being that there are no T's in mRNA. So you're going to go from A to T, but the T's are going to be U's. So it's going to go A to U, C to G, G to C, T to A. That one's still the same. That's what confuses a lot of people. That was transcription. That part's pretty simple once you understand it. The biggest thing, like I keep saying, is that people will usually mix up if they're supposed to use U's or T's. So just make sure you're on the lookout for that. What we do with that information, which we're going to do this week, is we're going to take that information that we got from transcription, we're going to take that mRNA code, we're going to turn it into a usable protein, and all you have to do is take that sequence that you got and plug it into an amino acid chart that will tell you what amino acid each code codes for, and then just tell me what it codes for. So it's really just a matter of looking at the mRNA sequence that you have, and then looking at the chart and telling me, okay, this is what the chart says my thing should code for, and we're going to do some practice problems today. So for today, there's not going to be actually anything that I'm going to have you guys do on your own yet, because I want to get a chance to explain it first, do some practice together, and have you guys ask questions if you have them. And then tomorrow and Thursday, we're going to do some practice where you guys are going to translate mRNA sequences for me, and we're going to see how you guys do with that. So let's do some practice. Let's say we have a DNA strand. So this is the DNA strand. We have not transcribed it yet. I picked a short one to start off with. We're going to do another one in a second that's a little bit longer. AAA, GCA, TTC. Now remember, now that we're doing transcription, we're not going to be able to use T's in our new sequence because mRNA does not have T's in it. So for every time we're going to make a T, it's going to get swapped to U. So if it helps you, I know I keep saying this, but if it helps you, you can do this the same way you'd been doing replication. So AAA would go to TTT, and then just do it all normal to start with. And then once you have your whole sequence, go back through and just change all the T's to U's. But if you can understand just flipping, going straight from A to U, that works too. So here's how this is going to work. So AAA is the first sequence. Normally, like I said, that would go to TTT if we were doing replication, but we're making an mRNA sequence, and there are no T's in mRNA. So that's going to be UUU. G goes to C, C goes to G, A goes to T again, but T's go to U's. So C, G, U. T still goes to A. That's what still confuses a lot of people. So even though A no longer goes to T because there are no T's, there are T's in the DNA sequence, and the T's still go to A's. So this is going to be AAG. So here's what it would look like, and I'll leave that up there for a second. You can also pause the video if you need to. So again, A will go to U for mRNA. G will go to C, C to G, A to U, T to A, and C to G. So you've got UUU, CGU, AAG. This is the same thing that we did last week. This is just transcription. Honestly, I think this is the more difficult process of the two because of the rule between U's and T's. Once you have this, hopefully the next part will be really, really easy because all you have to do is take this information, plug it into a chart, and just tell me what the chart tells you. So here's what I mean. Here's our sequence. And I mentioned this last week, but I'll say it again here because it's important. I will always give you this. I would not expect you to memorize all of this. I don't have all of this memorized. I could only tell you probably a handful of them off the top of my head without looking at the chart. So I will never ask you to memorize this. I will always give this to you. Or there's another version that I'll give you that some people prefer. I've got two variations of it. But anyway, all you have to do, once you have your sequence, all you have to do is plug this into the table. So our first sequence is UUU. You're going to find that on here. Right here, UUU. Codes for phenylalanine. They're all abbreviated PHEs. I think I said this on the next slide, but again, I will not ask you guys to know the full names, especially now that we're doing this through distance learning. I don't expect you to look up all these and know what they code for. I don't honestly know what all of them code for off the top of my head, like the ILE. I don't remember what that one. I think it's isoleucine. I don't remember. Either way, you won't know what these are named after because they're all abbreviated, and I'm not there to tell you directly what they are. So just put the abbreviations, and that's fine by me. So we've got UUU, which is PHE. Next one is CGU, which is over here. It's arginine, ARG. And then you've got AAG, which is right here, lysine, L-Y-S. Big thing that I see a lot of people making mistakes, and this is just a simple mistake that has to do with how our brains work. A lot of people sometimes will get letters mixed up, so you'll see AAG here, but some people get that crossed in their head when they go to look at the chart, and they'll think, okay, AGG, that's, well, yeah, that'll go for arginine, and it's just a simple letter swap, which, so I get some people that ask me, like, how did I get this question wrong, or why is my thing not making sense? Go back and double check. Sometimes you just swap the letter. I even did that when I was typing this out, so just make sure you're double checking either way. But like I said, this one hopefully shouldn't be too hard. You're just taking this information, plugging it in this nice, neat chart that a bunch of scientists were nice enough to make for everyone, so we don't have to figure that out on our own, all that jazz. So yeah, here's what it would look like. A couple things. First and foremost, I already mentioned all the amino acids are abbreviated on the table. I won't expect you to know what they are. Just put the abbreviations, just do what the table tells you to do, especially since we're not in the classroom, and that should be more than satisfactory. Second thing, if you noticed on this chart, there are a couple ones that say stop. Those are called stop codons. They're exactly what they sound like. They tell the DNA where to stop coding because, again, genes can't go on forever. Your DNA has parts where it starts and stops. A sentence has an end point where the period goes, and then there's no information past that point. Normally in the body, what this will do is this will cause the protein to stop coding, and then the protein can form and do its job. For our purposes, if you ever run into a stop codon, you can stop. You can end right there. Even if there's like 20 more sequences past that point, if it says stop, you can stop coding because that's how it would work in your body. You have a lot of DNA that doesn't get coded in your body because there are stop codons in between them. So you have big chunks of information that don't actually do anything because there's nothing to read it. So even if, like, say for example, going back to this example, let's say CGU coded for stop, even though we've got this other one right here on the end, even though this exists here, since we stopped here, you don't have to read it. If you go on ahead and code for it anyway, which a lot of people do just out of habit, that's perfectly fine. But if you're doing an assignment and you come to a stop codon and you stop there, I'm not going to count you off for that because that's how it would work in your body. And the last thing, like I said, I will also link, there's another version of this I think I showed on a different video, I think on the PowerPoint from last week. There's also a wheel version of this where some people visually like it better because I think some people, depending on how your brain works, some people can use it a little bit easier. But I'll always link both. It'll be up on today's Google Classroom notes too, and I'll post it for the next couple of assignments as we go forward too. And I'll always give that to you, like I said. So let's do one more example because this is going to relate to something that I'd like to do tomorrow and Thursday. So we've got a new sequence here. GTT, GAG, AAA, TGC. You get the idea. I'm not going to read the whole thing. First step we have to do, this is DNA again, so now we have to decode this into mRNA. I'm just going to show you the end result so I don't have to waste a lot of time saying it all out. Again, just double check yourself, make sure this makes sense. All I'm doing here is I'm matching letters. G's go to C's, T's go to A's, C's still go to G's, and then A's will go to U's because there are no T's in mRNA. So here's my sequence. Pause the video if you want to double check, make sure I've done everything right. I think I have. I've looked through this a couple times and I'll look through it again right now. Okay, so here's what we're going to do. All you have to do is, again, if this was your sequence, you'd go to the table, you'd look for CAA, go down here. That is right here. GLN, I think that's glutamine. I forget. There's a lot of ones that sound similar. Going to the next one, CUC, that codes for leucine, phenylalanine, ACG, where is that? Right there. Threonine, I think is that one. I forget. Either way, that's all you have to do. You have to plug in chugs. So for this week, because I realize that's really tedious and a lot of it is kind of abstract because you don't know what these amino acids are. You probably don't even know what these words stand for. They're just a bunch of letters. So to put this into a better context, to make it easier to understand and also way less boring than going to a chart and filling out a bunch of chemical names, here's what we're going to do instead. So normally this is what it would look like. That's really boring to look at. We might do something more like that just for the sake of being thorough if we've got time, but to kind of make this a little bit more enjoyable and a little bit easier, we're going to do it this way. If I can find out where I put it. Here we go. So this activity is essentially the same thing as what I just showed you, but instead of coding for amino acids, we're going to code for letters. This way I can make a secret message that you will uncode, and it makes it a little bit more accessible than if you were just like plug in arginine, phenylalanine, all that stuff. So I'll show you what I mean. Here's my sequence here, and here's my table, and I'll give you this table when we do this activity tomorrow and Thursday. So here's my code that I made for today. This will not be the same code as tomorrow, so don't get your hopes up. It's the same code I just showed on screen, but instead of coding for amino acids, it's going to code for letters. So I'm going to go to my chart. First one's CAA. That codes for, let's make this a different color. CAA codes for, where is it? T. CUC codes for R. There you go, translation, if I did this right. So hopefully that's what you got. If you're confused about anything, let me know. That's what the purpose of today is. There will be one Google form question for today that I'm planning on doing, just to make sure that you watch this video and that you understand everything. The main thing will be like, what is my code? So I know that you watched the video. The code is translation, so that'll be the answer to today's Google form question. And then I'll put a second box just for any questions if you have them. That way I can look at them and be kind of prepared for what to expect for tomorrow, and if I need to explain anything a little bit better. That will be optional. If you don't have many questions about this, you don't have to fill it out, but I'll put it there just in case. So anyway, that's it for today. Make sure you're ready for tomorrow. Again, it's going to be this. This is what I'm planning on for tomorrow. I'll make a new code. I might do a couple, depending on how time-consuming I think they'll be, and how in-depth I want to make it. But either way, I'll give you some new codes. You should be able to tell if you did it right, because if they make words that make sense, then you did it right. If it's a bunch of gibberish that doesn't mean anything, then something probably went wrong, which at that point you can either redo it or just email me and I can tell you what you did wrong. But either way, that's it for today. Let me know if you have any questions, and I'll see you guys tomorrow.